Hi, welcome to our presentation on neural echoes. My name is Zahra Babaiee and I'm going to present that depthwise convolutional filters replicate biological receptive fields. CNNs are heavily inspired by the visual system. The visual system consists of two main parts, the retina and the visual cortex. The retina is the first processor of the images, so the visual information lost in the retina can never be recovered. Many retinal cells, known as ganglion cells, have receptive fields with center surround structures. These cells either have excitatory centers, meaning that they get excited when light falls on their centers, or the other way around, with inhibitory centers. Difference of Gaussians or DOG is a common function used in neuroscience to model the center surround structure in receptive fields. Since AlexNet, it is known that the first layers of CNNs display Gabor-like patterns. However, as we go deeper, the kernels lack clear, understandable patterns. Here you can see random samples of kernels from all layers of three widely used CNNs with regular convolutions. Depthwise convolutions, however, exhibit a fascinating characteristic that we have uncovered. These convolutions, unlike regular ones, reveal a pattern of repeating structures in their trained kernels. This slide showcases a variety of random kernel samples from different models, ranging from MobileNet with 3x3 filters to those with larger kernels like ConvNext. Notably, a majority of these kernels demonstrate a center surround configuration, with both excitatory and inhibitory centers. To validate our observation of center surround structures in most kernels, we use the k-means clustering algorithm. We categorize the kernels into three clusters, one each for excitatory and inhibitory centers, and the third for other patterns. This slide displays random samples from each cluster alongside the cluster's average kernel at the bottom. Notably, the first two clusters align with the excitatory and inhibitory centers, whereas the third cluster, representing diverse patterns, results in a more cluttered average representation. In this slide, you can see the bar plots of the number of kernels of each cluster for different variants of ConvNext v1 and v2 models. ConvNext v2 is the improved version with higher accuracy. Interestingly, we observed that the number of kernels from the center surround structure increases in this enhanced model. Here you can see the bar plot of the filter cluster proportions from different variants of various model families. It is notable that in each model family, we observe almost consistent proportions of filter clusters across variants of different sizes, kernel sizes, and even dataset sizes. Inspired by our discovery, we propose a center surround initialization method for depthwise convolutional kernels. We use the neuroscientific model of DOG, and given the kernel size and the ratio of center to surround, we can calculate the variance as given. As shown, the produced kernels are very similar to the depthwise convolutions, mostly with a small ratio of center to surround. Here you can see the results of ConvNext, Hornet, and ConvMixer models trained for 50 epochs with widely used k-means initialization compared to our method. Our initialization improves the accuracy of the models up to more than 2% for the ConvMixer model. We also performed an ablation study on the initialization parameters, which revealed that a small ratio of center to surround and having both excitatory and inhibitory kernels works the best. To summarize, we present the discovery linking artificial neural networks and natural vision systems through center surround patterns in depthwise convolutional kernels. Substantiating this evidence, we provide analytics of trained kernels from various state-of-the-art models. Finally, we introduce a novel initialization strategy for these kernels inspired by the difference of Gaussians method used in biological vision models. Thank you for your attention.